Basically, our program, I see it as a link between the clinic and the basic research side of things. We collect tissue samples and blood samples, other body fluids from patients who have cancer, and we process them in specific ways. We store them in the freezer in specific ways, and then when a researcher, for example, is looking into melanoma or lymphoma, they'll ask us for tissues, and then we provide them with those samples so that they can sequence them or look at the different types of abnormalities that might be there compared to normal tissues. So every patient who has a tumor has an owner at the end of the leash. The owner is the one who actually gives us permission to collect that piece of tissue or that serum sample, and then lets us know that it's okay for us to use it how we think is most appropriate to advance cancer medicine. Some of the things we do are to help discover about cancer. Why does this happen? Why does this not happen? Is it the same as humans? The idea ultimately, if we can know how cancer behaves in both species, is can we then translate it back to a treatment perhaps? If we find something that's wrong with this particular gene, can we figure out a treatment or take advantage of something they're studying in humans and then adapt that back to our veterinary patients?
The biorepository is kind of just a priceless resource for us, because when we get ready to do an experiment, we contact the administrator for the biorepository and we basically ask, for this tumor type, say lymphoma, what kind of samples do you have available for us to use? And then we can pull those tissue samples or those blood samples out of the biorepository and prepare them for the types of experiments that we do. Now in our case, that's usually isolating DNA and RNA from those samples. So DNA and RNA are the codes that tell cells how to behave. And in cancer, there will be mistakes in that code that allow for unregulated growth of the cancer. So we're doing all of this sequencing so that we can identify those mistakes, so we know what's causing those cancers to have the unregulated growth. The other thing that we've been doing is we've been taking some of those tumor samples and we disperse the cells and we grow them in culture. And so this gives us a continuous source of those cells so that we can test new drugs or new therapies in those cells prior to putting them into animals.
Some specific examples of how our tissues are then used in a basic science way. To use Dr. Duval's lab again as an example, she had a project where she and her graduate students were looking at canine bladder cancer and looking at the molecular markers to see if it was similar to what they see in people as far as the different mutations that are seen, and to try to find out what drives canine invasive bladder cancer.
So one of the big handy things that we can do is we can compare the mistakes that we find in the canine tumors to mistakes that people know are important in human tumors. With that particular mutation, there have been drugs that were developed that would specifically target that mutation in human cancers. So one of the first things we do is we go into our cell lines that we have and we look for a cell line that carries that same type of mutation, or maybe we can even generate a cell line that carries that same type of mutation, and then we can take those human drugs and we can test it in that cell line to see if we get similar responses that they've seen in human cell lines.
My goals for the biorepository going forward, one thing I'd like to see us focus on is how are we gathering the patient outcome information to match with those samples. That makes the samples even more valuable.
You know, it's been very critical to be able to partner with Dr. Lana, because when we can partner with her, then we can make sure that as the biorepository is collecting samples, that they're collecting the types of samples that we really need the most, and that they're being stored in the conditions that we need them for our future experiments. So that's been a critical aspect of our research. And so my hope is that by going through and really teasing apart the code that drives these cancers, that we'll be able to identify therapies that are far more effective.